Hello, everybody. Welcome to today's webinar, which is the first one in a series of events planned by Femtonics for the coming months. I am Esther Marco, product specialist at Femtonics, and I also have Dr. Dana Spafi here with me today, who is the head of our application specialist department, and he's going to help answer your questions about today's topic, which is smart solutions, smart microscopes. With this series, we would like to support the neuroscience community through the COVID-19 period by supplying you with useful information regarding two-photon technologies and organizing one-to-one demonstrations for those who become further interested. The series will cover a wide range of topics, from the benefits of conventional Galvo and resonance systems to advanced applications like imaging dendritic signals using acousto-optic systems. Please note that although you will be muted during the presentation, you will be able to use the Q&A option at the bottom to submit your questions, which will be answered after the presentation. In case we run out of time before we get to your question, we will make sure to send you our reply in email to the email address you used to register for this webinar. With that being said, let's jump to today's presentation, which I recorded for you in advance in case any unforeseeable circumstances arise.
Enjoy.
Today, we will start with a short introduction of Femtonic solutions and discuss why it is important to have a stable and modular microscope design. Not only are we going to highlight the benefits of Galvo-Galvo, Resonant-Galvo, and dual systems, but we will also be showing images for each to help better understand the possible applications. We will also touch upon why Femtonic solutions are unique. And lastly, we will be finishing with a Q&A session. To put conventional Galvo and resonance systems into perspective, I'd like to briefly outline the Femtonics portfolio. Just like how the acousto-optic technology is the most advanced solution currently for true 3D imaging, Fem2 Atlas, our product utilizing this technology, is the peak of the evolution within the Femtonics portfolio as well. Because Fem2 Atlas bears with not only the imaging functions provided by traditional Galvo and resonance scanners, but also combines these with a unique fast 3D imaging feature, it can be viewed as an all-in-one tool. The acousto-optical technology, together with Atlas, will be the subject of upcoming webinars, and now we will proceed with introducing the Smart product line. Scientists with a more modest budget, or those who are more comfortable with getting acquainted with two-photon technologies gradually, can rely on the FEMTOSmart product line with Galvo or resonance scanners, or both. Another advantage of the FEMTOSmart product line is that it is highly modular and can be customized to specific applications both on the hardware and software level. FEMTOSmart was designed with keeping stability, high signal-to-noise ratio, and precise control in mind. Extreme positioning freedom is granted by a built-in microscope body positioner, which creates a 50 mm moving range along the 3 axes with a 1 micron minimum step size for easy navigation. The objective is 360 mm above the optical table, and the system can be mounted even higher as a custom solution. As mentioned, the product line is highly modular, which allows it to be fitted to various research needs. We will discuss these later in this webinar. Femtonics microscopes utilize a so-called traveling detector system, meaning that the PMTs are as close to the objective as possible at all times, even throughout imaging, to eliminate any potential signal loss. And while we're discussing hardware features, I'd like to highlight that resonance systems at Femtonics come with a Pockle cell, and you do not need to procure one separately. Not only can the FEMTOSmart product line be used for in vitro experiments, but the elevated and well-balanced body provides ample space for in vivo studies as well, even with behavioral devices attached. Looking at our setups in our customers' labs, the range of model animals they use is just as wide as the range of behavioral devices that we have integrated for them using analog or digital channels. For example, we have customers with small model animals like fruit flies and larger ones like non-human primates. Behavioral devices we commonly integrate include gramophones and other head-fixed designs combined with virtual reality, all developed by Femtonics, a wide range of Lewis and Neumann devices including treadmills, Phenosis jet balls and accessories, and mobile home cages from Neurotar. From the laser to the sample, the optical light path is simple and customizable. Usually, the transmission range is requested to be between 680 and 1100 nanometers, but it can be extended up to 1700 nanometers. The microscope body houses the scanner unit or units, the internal light path, and also the control circuits. It was designed so to keep electrical signals isolated and avoid the antenna effect. The hardware is the same for Galvo and resonance systems with a difference in the scan heads. There are separate light paths for both, and they can be used simultaneously as a dual system combining imaging with photostimulation or dual color imaging. By default, the microscope comes with 2 non-descanned gas detectors, but it is able to hold more. Our system is equipped with the fastest galvano scanners available on the market today. It allows the scanning of a 512 by 512 pixel area at a 4.1 frame per second speed. As opposed to resonance scanners, Galvo scanners enable flexible scanning patterns. These built-in ROI scanning patterns increase signal-to-noise ratio, which is further improved by high-quality detection optics. The hardware is controlled by our MATLAB-based software called MES, which provides intelligent control while remaining easy to use. Now let's look into the primary applications of a Galvo scanning 2P microscope. Line scans are typically done by Galvo scanners. Note that the scanning is in fact always continuous, but the software interprets only the signals coming from your defined patterns as meaningful, which happen to be lines in this case. Let's imagine you're imaging a network of neurons in the mouse cortex, as seen on the left side of the slide. By laying down one line scan pattern per soma, marked with numbers on the leftmost image, if there is no major movement, you're able to read out their individual activity with high acquisition speed and compare them on a heat map. At the bottom, the same delta F over F is presented over time that was marked with red on the heat map above it. We use GCAM6 labeling, and you can clearly see nice calcium spikes throughout the 5 seconds of this measurement. On the right side of the slide, marked with blue, yellow, and red, you can see that you can also apply line scans to acquire data from different segments of a dendritic branch. These correspond with the calcium response curves on the right. We use GCAM6 labeling here as well. Femtonics was the first company to develop commercial software solutions for special area scans. We've developed patterns to follow and reasonably oversample around a region of interest, in this case here, a proximal dendrite. Should the sample move to any natural causes, we're able to correct for motion artifacts, allowing us to collect realistic calcium measurements from the dendrite. Another useful application of Galvo scanning is uncaging. On this slide, you can see images from a mouse brain slice preparation where we used a caged form of glutamate developed by Femtonics called DNI glutamate, and we uncaged it by applying a specific Galvo scanning-based point stimulation pattern, which you can see in yellow. Then, also with Galvo scanning, we collected calcium responses using 3 sequential line scans along the dendrite, depicted with green, orange, and blue arrows. To validate data, we also used electrophysiological patch clamp measurements. We can do this uncaging experiment with 1-photon continuous wave lasers or with 2-photon lasers if we are in deeper tissue. Galvo scanning and the stability of electronics also allows us to acquire detailed images on a longer timescale, as in this example of the developmental study of a zebrafish embryo. The MES software offers various programmable protocols enabling different measurements to be performed sequentially, even for hours. Another feature which provides flexibility to investigators is that you're able to control the imaging session remotely as well. As mentioned, we're using the fastest Gallo scanners available today, but their speed is a limitation if one would like to expand the region of interest to a full frame. In this case, resonant scanners can be used, enabling faster sampling. Resonant mirrors resonate and guide the laser beam at a given frequency, which is 8 kHz in most cases. This means we can acquire a 512 by 512 pixel area at a 31 frame per second speed, which can be extended to a given volumes with the help of a piezoelectric objective positioner. An intuitive graphic user interface helps the user to record their in vivo measurements and obtain excellent image quality. As said, Resonant FEMTOSmart systems come with a Pockle cell to provide fast control over laser intensity to minimize photo damage. Here are the primary applications of a resonance system. On the left side, thanks to G-Camp injection, you can see a beautiful network image from a mouse visual cortex. There are some cells which overexpress G-Camp, and there are plenty which have the desired donut shape. The image is 512 by 512 pixels and shows a 200 by 200 micron area. On the right side, we're looking at a volume scan of 50 micrometers thickness. Researchers can use fast piezo drivers or liquid lenses to record volumetric images fast enough to see calcium transients. In this case, we used a 100 micrometer travel distance piezo objective positioner to scan through this volume of the mouse visual cortex. So far, we have discussed the benefits and applications of Galvo and resonance scanning when used individually. In a dual system, the Galvo and resonance scanning modes are combined, and you can switch between the two from experiment to experiment. Moreover, if you have two laser outputs and the two light paths have been combined with a dichroic mirror, you are able to perform simultaneous imaging and photostimulation. As mentioned, you can select different stimulation patterns by the Galvo scanner, and on a different imaging wavelength using the resonant light path, you can read out the direct and indirect effects of photoactivation. In the microscope body, the resonance scanner is above the Galvo scanner, and the two light paths are joined by a dichroic mirror that has a wavelength cutoff customized to your application and the dyes you plan to use. The animation shows nicely how one can imagine the simultaneous work of the two scanners. And here you can see a real-life application. In the video on the left, 3 neurons were activated by uncaging glutamate with a Galvo scanner at 740 nanometers using point stimulation, and imaged the process by simultaneously scanning with the resonance scanner. On the video on the right, we did the same, but with 2 spiral stimulation patterns and on a bigger area of the radiatum and CA1 region. By using an optogenetical background or uncaging, we're able to manipulate cell and network activity by deliberately activating or inhibiting the region of interest. To imagine how this is solved on a hardware level, please investigate the schematics of the FEMTOSmart dual system on the right. If there are 2 titanium sapphire laser beams available, we're able to guide both laser lines through both light paths, controlling the intensity with a Pockle cell in each light path. By doing this, we're able to scan through the sample with 2 different colors, resulting in dual color images, where not only the detection is based on 2 different channels, but also the excitation happens at 2 different wavelengths. There is a wide range of modules available to support different applications, and with some exceptions, this is independent of the scanning type. This makes our system preferential for many research questions. Of particular interest to our customers are in vivo green illumination for vessel pattern visualization, the LED light source for full field illumination, and modules that enable free positioning, such as the XY body positioner, which moves the entire microscope along the X and Y axis, the motorized tilting objective unit, which introduces virtual axis for the positioning of the objective, and the so-called bridge structure, which opens up space under the objective even further if, for example, you have large animal models and/or behavioral devices. The in vivo green illumination module helps orientation on the brain surface by visualizing blood vessels. The video on the right was taken by a CMOS camera. Even the thinnest blood vessels can be seen, which enables you to find exactly the same location during chronicle experiments. The system is easily convertible for in vitro studies by introducing an in vitro platform. It transmits illumination at 840 nanometers, uses gradient contrast, and includes an oil condenser with an Na of 1.4. It can also hold substage PMTs and PMT or photodiode detection units for the transmitted infrared light to study further anatomical questions. As one can assume, it can be surrounded by manipulators to perform electrophysiology parallel to imaging. Converting the system between the in vitro and in vivo modes, or vice versa, is fairly quick. You only need to screw the in vitro platform on or off from its fixed position under the objective. Of course, you will need to set the transmitted infrared light path for optimal illumination and contrast. This way, one system can be used for any animal model in vivo and also for tissue sections from their organs. Infrared illumination, as part of our in vitro extension kit, is also a classical but useful method. On this example, it is used for patching a cell, showing the momentum when the glass pipette attaches to the neuron. Through a CMOS camera, you can see fine details like the small depression caused by the pressure from the pipette. We are very proud of our unique solution designed for reaching brain areas that are not parallel to the objective's plane. The motorized tilting objective model rotates the objective 180 degrees around the horizontal axis and 100 degrees around the vertical axis, giving it a higher level of freedom to reach the sample from different angles. The module also includes green illumination, which we've discussed just now, and a piezo objective positioner, which enables additional movement in the Z direction. The system is capable of high precision movements to fine adjust position and of high precision alignment, eliminating the need for touching the assembly, and you can store and reuse optimal angles and positions when experimenting multiple times with the same animal. It is suitable for imaging lateral brain regions like the auditory cortex, even in non-human primates. On the picture, we can see an anesthetized mouse experiment while we record from the auditory cortex. As proved by the Z-stack in the middle, the system is able to produce high-quality images along this alternative axis as well. In addition to the tilting objective module, there is another positioner available which fulfills a more basic need: positioning in X and Y. The FEMTOSmart XY body positioner is built in the column of the microscope and provides a 50 mm moving range with a 1 micron minimum step size. Both the tilting objective and the XY body positioners can be controlled from the software and alternatively by a handwheel as well. The bridge structure was designed to adopt special experimental conditions with large samples and accessories. The stability of the optical path is maintained throughout the whole travel range in Z, which is 500 millimeters, and the motors have memory positions which saves time during chronic imaging experiments. The design is compatible with all modules discussed in this webinar, but most often it is equipped with a combination of piezo and tilting objective positioners for volume scanning with extended flexibility. We have successfully installed the smart bridge structure at various labs around the world, including the prestigious Norwegian University of Science and Technology. Full field illumination using a selected LED source allows molecules and cells to be stimulated over the whole field of view homogeneously. This is a cost-effective, easy-to-use approach mainly used for optogenetics. The wavelength of the LED can be fitted to customers' needs as well. We combine this module with gated detectors to achieve microsecond switching between stimulation and imaging. While the LED light is on, the gating of the PMTs are automatically activated to retain the PMT sensitivity on the long term. Since light pollution can be a disturbing factor, especially during in vivo experiments, we've developed other methods as well to enhance signal-to-noise ratio. In a FEMTOSmart dual system, a one-photon continuous wave laser can be coupled with the primary light path and can perform all the flexible stimulation patterns mentioned earlier, either sequentially to or simultaneously with two-photon imaging. The main applications for one-photon stimulation are uncaging and optogenetics. Usually in vitro, since the penetration depth of visible light is limited to a few hundred microns, but of course, this does not mean it could not be used for in vivo samples when the fact of reduced penetration is not hurting you in reaching your experimental aims. Another advantage of 1P photostimulation is that it is a less expensive approach than two-photon stimulation, and similarly, multiple wavelengths can be chosen in this case as well. We've discussed the hardware requirements and applications of two-photon stimulation extensively earlier in this webinar. It provides advantage over one-photon stimulation by enabling deeper penetration and provides advantage over LED photostimulation by having better spatiotemporal resolution. For these reasons, two-photon stimulation is usually used in vivo settings. We've discussed quite a lot during this webinar related to the FEMTOSmart product line, and there are two new items which I'd like to bring to your attention, namely 3-photon excitation and fluorescence lifetime imaging. 3-photon excitation is gaining increasing attention since longer excitation wavelengths cause less scattering in biological tissues, which comes with several advantages like increased penetration depth, reduced out-of-focus excitation, and in turn, increase signal-to-noise ratio. At Femtonics, we will fulfill this rising need by unlocking the option to provide FEMTOSmart systems with 3-photon range optics with a transmittance between 1200 and 1700 nanometers. The figure on the left shows fluorescent activity measured on several somata from the V1 region of a GCamp labeled mouse. In addition, we've established a long-term collaboration with Becker and Hickel, whose detector and software can be integrated into our system, enabling researchers to acquire fluorescent lifetime data along with fluorescence amplitude measurements. And on the image on the right, you can see a nice example of the normalized total count technique to map intracellular calcium in the plane of view. This example was captured by Zeng et al. From University College London, and we are planning to extend our collaborative network in this regard to work on new FLIM approaches. Now that we are reaching the end of the presentation, I would like to recap the key strengths of our FEMTOSmart product line. Simultaneous imaging and photostimulation is possible with a dual Galvo and resonance system. A wide range of additional modules are available to customize the final setup to your specific application. And it was designed to grant researchers with enough space even for in vivo studies with behavioral devices and provide a high level of positioning freedom. And finally, let me mention the topic of our next webinar. In the beginning of this presentation, I highlighted the two main product lines of Femtonics, one being FEMTOSmart, which we've discussed thoroughly today, and the other being Femto3D Atlas, a product which uses a novel approach, namely the acousto-optic technology, to perform ultra-fast, random access true 3D imaging, in addition to being capable of what the conventional Galvo and resonance systems allow. Thank you so much for your attention. We will now proceed with the questions. Do not get discouraged if a question came to your mind just now, or you did not hear it now, as all of them will be answered in email.
Okay. Let me see. Thank you for being so active. We've received quite a few questions. Let's see. Okay, I think we should start with this. Pretty easy. What is the depth that can be reached with a FEMTOSmart system?
Well, I think it depends on many parameters. For example, the sensor and the objective, and of course the surgery technique. But I can say that in case of cortex imaging, you can go up to layer 5 neurons. It can be easily seen. And and in special cases, you can go down up to almost 1 millimeter with this technique. So it's, for example, if you are looking for some hippocampal imaging, when you remove the cortex and replace with the cylinder, then you can reach even the stratum radiatum, for example.
Thank you. On to the next one. Is it possible to combine tilting objective with piezo z-axis on FEMTOSmart? Yes, it is possible. In the tilting mode, you can select the option to perform z-stacks with the piezo orthogonally. And we have many users who are combining these positioners together. Yeah. Moreover, usually if someone uses a tilting objective model, they use it together with a piezo objective positioner. And it's something that is... it's quite useful when you want to do some fast function imaging. For example, if you are working with the auditory cortex, you have to move orthogonally in the virtual z-axis. With the objective, and then you can do really nice analytical z-stacks and functional imaging as well. It's for sure, it's very easy to combine them. Thank you. Okay. Do you have presence in the United States? Yes, absolutely. We are there for US customers 24/7. Regarding both technical questions and also application-related questions as well. We've been present at US conferences, I think for the last 10 years. We've been there at SFN. And also, currently, we are actively expanding as well. And we have demo labs in the United States. After the COVID-19 situation, you are welcome to check out our systems at these demo sites as well. Okay, next question. I have my own MATLAB scripts. Can I use them to control the microscope?
Yeah, of course. Yes, the answer is yes. If you have some nice scripts for analysis or something, then you can easily run on your MATLAB code after the measurement, or even you can control the microscope as well from the command window. Yes.
Thank you. Next one. I have a lot of motion caused by animal movement. What solutions can FEMTOSmart offer for that?
I always advise that I think improved the surgery technique is the best solution to reduce the movement artifacts. But in case of if you cannot avoid this, then you can use rigid and non-rigid motion correction as well after the measurement. So there is a built-in function in the software for this reason. And I also prefer some open source platforms. So it could be very, very useful because our format, HDF5, it's supported by many other platforms as well. So if you prefer other techniques, then you can use it.
Thank you. Next one would be, how easy is it to switch between resonant and Gallo scanning?
Oh, okay. Yeah, it's quite easy. It's only one click on the GUI. Yes, everything is automatic, so you don't have to touch the rig. You can control everything from the software. And if, for example, if you want to do some nice anatomical images with the Galvo-Galvo, then you can do. And after that, just after a second, you can start your function imaging as well with the Galvo Resonant. And moreover, you can programmable this as well. So you can build really nice protocols to repeat in a loop mode all of these things. Yeah.
Okay, thank you. Yes, the presentation will be available on YouTube.
Nice.
Yeah.
And another question there, is it possible to combine Titan Sapphire laser for imaging and one-photon laser for uncaging on FEMTOSmart? Of course, yes. I think it was in the presentation as well. You can order CW lasers as well for the microscope, for the rig. So if you prefer one-photon excitation for uncaging, then it's possible. I do recommend two-photon uncaging for several reasons, but of course, in special cases, it's also quite useful, the one-photon as well.
Thank you. Okay, yeah, I think this question concludes our Q&A session. Thank you very much for being here with us today. We appreciate your time and interest. Again, if for some reason we missed your question, we will get in touch with you in email after the webinar. And in addition, we will be sending out a short questionnaire to collect your feedback, which is warmly welcome because it is our first webinar. We hope you stay with us for our future events. The next one will be dedicated to discussing the novel acousto-optic technology and our product line which utilizes it, called Femto3D Atlas, which I mentioned in the presentation. There will also be a separate event that introduces all the different applications the technology enables. And we will also dedicate a separate session to introduce the software. I think that's it. Once again, we hope you enjoyed today's webinar by Femtonics, and thank you for your attention.
Yeah, thank you very much. Bye-bye.